My name is Kasey Hobert, and I'm a senior pre-veterinary medicine and animal biosciences major with a minor in equine science here at the University of Delaware. This summer, I'm working with Dr. Amy Biddle in the UD Envision program on my project, which is community profiling of protozoans within the equine microbiome. Protozoa are present in the equine gastrointestinal tract and work synchronously with the bacteria to contribute to starch digestion during digestion. We currently do not fully understand what causes the community changes or how that change affects the overall function of the hindgut. This summer I worked on summarizing different protozoal communities found within the horse's gut based on factors such as differences in farm locations, breed, diet, and the body condition score of the animals. Protozoa communities were evaluated using fecal samples from horses, and DNA was extracted for genetic sequencing. This is the same technique that we use to characterize the bacterial community in the gastrointestinal tract, with minor changes for the protozoans. Communities can be evaluated with a number of different techniques, but two of the most common methods include alpha and beta diversity tests. When looking at alpha diversity, you will typically see a chart like the one pictured here. At first glance, this can be a little disorienting to the untrained eye, so we are going to break it down using simpler terms. Alpha diversity analyzes differences found within a sample. For example, in the sample pictured here, there are a total of 6 shapes within the circle. The alpha diversity measure, observed species, looks at the total number of unique identifiers. So in this example, we can see that there are 2 green triangles and 2 orange crosses. Since there are duplicates, we will not count these 2 extra shapes, and we will count 4 unique shapes: the lightning bolt, the triangle, the cross, and the moon. So the alpha diversity of this sample would be 4. Moving to beta diversity, we can see that once again the typical figure used to represent beta diversity doesn't show a lot without background knowledge of this type of analysis. So once again, we're going to break it down. We're going to use the same example that we used to analyze alpha diversity and add a second sample to it. While alpha diversity looks at the different objects within a sample, beta diversity studies the differences in objects between 2 samples. For this example, we can see that there are both similarities and differences between the shapes in the samples. Beta diversity analyzes the difference between samples. Therefore, the repeated shapes found in sample 1 and sample 2 will cancel each other out, and we will count the number of unique shapes found between the samples. So, with 5 different shapes between the 2 samples, we will have a total beta diversity of 5. Alpha and beta diversities are great methods to evaluate the similarities and dissimilarities found between samples when analyzing the data to look for differences between horses and management factors. We hope that the results of this data will better describe the diversity and the importance of the protozoal community within the equine gastrointestinal system and the function of their digestive systems.